Dr. Patil?
Yes, sir, I am here.
Yeah, ready. Okay. Once again, I wholeheartedly welcome all the participants for today's 154th talk, 119th monthly talk. Today with us, Dr. B.L. Basavaprabhu Patil. Before we proceed with this talk on one of the important emerging viruses, emerging Emaraviruses, and urgent need for major emphasis. For the benefit of the audience, I will go through his biodata. He is very interesting. He has several fellowships. He traveled a lot. And since beginning, he has been working on viruses, plant viruses. Coming to brief introduction, presently, Dr. Basavaprabhu Patil is working as a principal scientist in plant biotechnology at ICR, Indian Institute of Horticultural Research. He did his BSc and MSc in agriculture from University of Agricultural Sciences, Dharwad, and he did his PhD during 2001 to 5 from University of Delhi. During PhD, he received a DART scholarship to work at University of Stuttgart, Germany. All of you must be knowing DART fellowship is from the Germany to the developing countries. And he did his postdoctoral research during 2006 to 2010 in Donald Danforth Plant Science Center, St. Louis, USA, and contributed to the development of virus-resistant cassava for Africa that has been approved for commercial cultivation in Kenya. Briefly, he worked as scientist D in the DBT's National Agri-Food Biotechnology Institute located in Mohali, Punjab. And then he relocated to University of Basel, Switzerland. And he worked with Professor Thomas Hahn in the area of plant virology in Switzerland. In 2012, he joined as senior scientist in ICR's National Institute for Plant Biotechnology, New Delhi. And after 6 years, that is in 2018, he relocated to ICR's Indian Institute of Horticultural Research, Bengaluru, and continues to work in the area of plant virology and biotechnological interventions for control of viruses. During his stint in the ICR, he has been the principal investigator for multiple projects funded by DBT, BIRAC, BCIL, and ICR. He has published extensively and is a recognized editor for several international journals, and he was a reviewer for several Indian and international grant applications. He has served as an external expert to assess the scientists from and CSR and other organizations. He was honored with the Fellow Award by the Indian Virological Society in 2014 and received EU's Erasmus Mundus Scholarship in 2016. Recently, he was conferred with Professor B.M. Johri Memorial Award by Society for Plant Research India. During last year, that is for 22-23, he is the only agricultural scientist from India to receive the prestigious Fulbright Nehru Visiting Faculty Fellowship to visit University of California, Davis, USA. And this is with this brief introduction. Now I request Dr. Basavaprabhu Patil to go ahead with his presentation on emerging Emaraviruses, an urgent need for major emphasis. Over to Dr. Patil.
Sir, can you give me the rights to share the screen?
Yeah, just I'll go this. Yeah, please try it now.
I don't know.
Your PPT is open, no?
Yeah, it's open. Yeah, it's not. Yeah, now I can see it.
Yeah, I gave for share. Yeah. Yeah, we could see first slide.
Yeah. Good morning, everybody. Happy to see the stalwarts of plant protection here in this particular session of AFME. This is my second talk in this particular society. I had given a talk when I had just moved to IIHR in 2018. Yeah. One thing what I can say is at least these virus names are more simpler to spell than my name. Probably I saw Dr. Sridhar struggling with my name, but he was quite comfortable in spelling out the virus names. So I'm sure everybody should be comfortable to understand the fundamentals of this virus. Thanks to Dr. Varghese sir for highlighting that biotechnology has not been really able to deliver as much as probably people expected. It's a new area of research. Biotechnology in human biotechnology has delivered a lot, but plant biotechnology, there have been a lot of other things. But I don't know whether should I consider myself a biotechnologist, although my designation is principal scientist biotechnology. I've been working in the area of plant virology, and my MSc master's is in genetics and plant breeding. Then I moved to Delhi University and in plant molecular biology department, but started working on viruses. Okay. Well, this Emara viruses, I don't know how many of you have heard this name in the past. I have been working on these viruses since 2014, the time after I moved to NRCPB or NI, National Institute of Plant Biotechnology in Pusa campus in Delhi. And that's because pigeon pea sterility mosaic virus is an Emara virus, which is a major problem here in India, and especially in South India. And I come from a place called as Gulbarga, or of late its name has been changed as Kalaburgi. That's the tur bowl of the world. That's the bowl of the pigeon pea where the highest area of cultivation and the best quality pigeon pea is produced there. But since several years, in addition to pod borer, the plants are severely affected by pigeon pea sterility mosaic disease. In case of pod borer, you at least get some yield, irrespective of the quality of the seeds. But in sterility mosaic disease, if the infection is in early stage, you would not really get any yield. So, and the virus spreads much faster than the pod borer. So, well, that's the area on which I worked while I was at NRCPB, and also during my visit to UC Davis. However, since it's not a mandate crop here in IIHR, I'm not working on that. But yeah, I still have a lot of interest in this class of viruses. So, briefly, I would like to highlight about viral diseases and its diversity and the amount of the economics. Tomato is the crop which is affected by more than 136 different viruses. And globally, there are in 2017, that is 5 years back, the estimate of yield loss was so the total production was 40 billion. Probably if you now compare the price 100 rupees kg, probably it's now 100 billion or maybe more. Vegetables, you all know, they are severely affected by several viral diseases. Pepper is affected by 49, melon by 46 different viruses, lettuce by 53, potato 54, and it's only cucurbits which surpasses the number of viruses that infect tomato. That's 153 in number. So these are some glimpses. You all have seen this. I really don't have to explain much. This is some viruses infecting tomato and chili, okra, zucchini, and these are the viruses infecting some legumes like beans, cowpea, and also pumpkin. And what you see here is a severely damaged field by a Tosco virus, that is peanut or groundnut bud necrosis virus. This is a picture from Bangladesh. you can see how severely the farm is damaged. And I'm sure during the COVID pandemic, you have seen much more severe damaged farms, especially from Maharashtra, which were affected by a cluster of more than a dozen different kinds of viral species. So these are the viral diseases of roots and tuber crops. And this is the these pictures are the viral diseases of fruit crops. Papaya, you see, plum, peach, apricot, and cocoa, banana, citrus, yeah. And these are some of the legume viruses. And this is the viruses affecting floricultural crops and also cotton. Some cereal viruses. Here in India, we don't have cereal viruses. There have been, except for a recent report of virus infecting rice from Northeast a few days back, and a few years back there was tumor virus. Otherwise, we don't have much cereal viruses. So all these viral diseases, are caused by a diverse kinds of viruses with entirely different genome organization. It could be DNA or RNA in double-stranded or single-stranded forms, or it could be positive sense or negative sense. Till now, whatever pictures I showed, they are mostly either DNA or RNA viruses, mostly positive sense RNA viruses. There was not even a single one which is negative sense RNA virus. But here in this particular talk, I'll be highlighting on emaraviruses, which are negative sense RNA viruses. So all these viruses are encapsulated in different forms and shapes, so produce different kinds of virus particles, and they are transmitted from plant to plant by different means and methods, mostly facilitated by insect vector transmission. And from cell to cell and across systemic transmission occurs by different means involving several different viral proteins and its interaction with host proteins. Okay, this is about Bunia virals, the order Bunia virals, or they are called as Bunia viruses. So emaraviruses belongs to this, tospovirus, tenuvirus, they all belong to this particular order. Even the most deadliest viral disease reported from Africa, more deadlier than COVID. There are some of them which, like Ebola virus, also belongs to similar category. They also have negative sense RNA virus. Negative sense RNA virus, I mean here, they first produced a transcript which is in positive sense, and that translates the protein required for virus proliferation. Whereas in positive sense, it doesn't have to produce a transcript, but it directly encodes for a protein which do the necessary activities for virus establishment and movement. So this class of virus is entirely different. Flu viruses are also negative sense RNA viruses. So, generally these viruses are multi-segmented. They have at least 3 segments. The larger one is called as lRNA, the medium-sized one is called as mRNA, and the smaller one as sRNA. They encode for replicase protein with RNA-dependent RNA polymerase, and the medium one for glycoprotein, because these virus particles, they have a membrane. In addition to the core protein, they also have a membrane on it. So this feature is something common to several insect viruses. So there is a possibility that these viruses could also proliferate in the insect body during their course of transmission from one plant to other plant via insect vectors. So whereas the small RNA segment encodes for several non-structural proteins involved in some of the important functions of plant. So this is a phylogenetic tree which shows this is a summary of all Bunia viruses. Many of them, they infect different insects or human beings or some animals. But here, what you see here in green is the Fimoviridae, which infect the plant species, but transmitted by insect vectors, where it may also replicate within the insect vector also. So these Fimoviridae, the family name is Fimoviridae. It's recently established family. And the name of the genus is called as Emaravirus. This is the only genus in this particular family, and it falls in the order Buniavirales. These viruses are generally enveloped. These glycoprotein, they form the viral coat or envelope. This is in addition to the viral coat protein. And within the virus particles, the 4 to 5 or 6 there are up to 9 segments that may be encapsulated within this viral coat protein. And as shown here in this picture with linear diagrams, the first largest one encodes for RNA-dependent RNA polymerase, which is essentially the replicase which helps in replication of the virus. And the second segment encodes for glycoprotein, third one for nucleoprotein, that is the coat protein, and the fourth one movement protein. And there could be additional segments which may perform different functions. So this is transmitted by a special class of mites, not the typical mites you see around, but this is eriophyte mites, which are microscopic in nature. You can visualize them only through a compound microscope. So they are generally 80 to 100 nanometers in diameter, the virus particles, and this is and they are double membrane bound bodies. So the first Emara virus to be characterized was European mountain ash ring spot associated virus. So that's why it's called as Emara virus. The first letter of each word is picked up, and that makes the genus name Emara virus. And these are the different kinds of eriophyte mites that transmit. They are all different species. They transmit these Emara viruses. So this is the first Emara virus to be characterized. And what you see here is the eriophyte mite entangled within the plant tissue. And these are the virus particles, double membrane. They have a double membrane bound bodies that are present in the plant cells. And all right. So several years back, this was the only virus characterized in this particular family. But of late, with advancement in virus diagnostic technologies and sequencing technologies, several new Emara viruses have been characterized. Currently, there are more than 24 different species of Emara viruses that have been well characterized. Some of them, they infect rose, fig, and some of them wheat. This is quite common in United States. Coffee, raspberry, blueberries, corn, or bird, and some of the ornamental tree plants in Europe and North America are also affected by these Emara viruses, and also some fruits like kiwi, pistachio are also affected by this family of viruses. So this is the global distribution of Emara viruses. You can see here, this is in different continents. In the African continent, it's only fig mosaic virus that has been reported. And whereas in South America, it's only the wheat infecting Emara virus that has been reported. Whereas in United States, it's the majority number comes from the rosette virus, and followed by fig mosaic virus and several other Emara viruses. And in Europe, the most of the temperate fruit trees and several ornamental trees are affected by Emara viruses. There could be more. Every year, 1 or 2 publications come up with a new species of Emara virus. Hitherto they were not characterized, but with advancement in next generation sequencing technology and it becoming more and more cheaper, it has enabled discovery of several new Emara viruses. So in India or Southeast Asia, it's the pigeon pistolity mosaic virus, which is predominant Emara virus, followed by certain incidents of rosette virus. And in Japan, there are other viruses. Japan and China, there are other viruses reported. Probably in India also, we have many Emara viruses, but because of lack of not much research or emphasis, they still remain to be discovered. So this is a table which lists most of the Emara viruses, their size, the vector, the mite vector, eriophyte mite vector, which transmits them. And the one that transmits pigeon pistolity mosaic virus is Escherichia kajani, and it's also called as Escherichia kajani Channabasavarna because he was the one to characterize this particular mite and showed the transmission of this particular virus. And there are different host species. I'll not get into the details. And the number of segments associated with this Emara viral diseases range. Minimum there are 4 segments, and there could be more than 8 or even up to 9 segments that are associated with this class of viruses. And each of them, they encode for different proteins I have already described. And RNA 5, 6, 7, 8, they encode for different proteins, whose function is to be characterized. And this is an RT-PCR, the PCR amplification of different MRI virus segments from different viral diseases. You can see how is the diversity and the diversity in the size and also the sequence of different MRI viruses. And this is a phylogenetic tree of the MRI viruses of family Fimoviridae compared with the toscoviruses or peribunia viruses or Secoviridae. So here you see all the MRI viruses, they cluster together, and their sequence is quite close to toscoviruses like tomato spotted wilt virus or groundnut bud necrosis virus. So this is phylogenetic tree by using different segments. Sometimes because of segment reassortment, the trees are sometimes complex. They don't show similar clustering pattern as seen with RNA dependent RNA polymerase sequence. Usually whenever we do clustering, it is done with RNA1 segment, which encodes for the viral replication factor, that is RNA dependent RNA polymerase. So this is wheat mosaic virus in US. It is it was generally observed in high plains. That's why it was also referred as high plain wheat mosaic virus. And it's quite devastating in US. And it was only just few years back, people were able to discover that it was caused by MRI virus, sometimes in association with some of the wheat infecting portiviruses. And this is also transmitted from plant to plant by the Areophid mite, a different kind of Areophid mite species. And here is the viral genome structure. And you can see they are all of different sizes, and they are named from segment 1, 2, 3 up to 8. And yeah, as I said, they encode for different proteins. And these virus segments, the 3' terminal and the 5' terminal, they have conserved sequences. And these conserved and complementary sequences, they form a double-stranded RNA, and that results in formation of a panhandle-like structure, and that gives stability to these viruses, and it protects from the nucleases such as RNases from destroying these viral RNA segments. And these conserved sequences are very key for amplifying these viruses and for further sequencing. So these are the sequences. You can see how well they are conserved at either end of the viral segments, and each segment has different sequence patterns, and that varies from viral species to species. So this is the domain structure of different viral proteins, MRI viral proteins. For example, RNA-dependent RNA polymerase has some pre-domains called as AF, A, B, C, D, E. I'll not get into the details of these domains, but they have a lot of, these domains play an important role in recognizing the RNA virus, and they replicate by using a cap snatching technology. That is, they chew up the cap on the 3' end of the viral sequence, and then, it takes up into the replication cycle of the viral RNA. And I'll really not, probably it may not be of much interest to the current audience, so I'll not get into the domain details of these viral proteins. So this is the alignment of different viral proteins. You can see how well all these viruses have conserved domains with several common functions. And when you do phylogenetic analysis using different segments of different MRI viruses, generally they form 4 different clusters. You can see, although the pattern of their clustering is different, but generally they form 4 different clusters. So this tells how these viruses have evolved systematically, in response to the host species or the environmental factors. So I'll not get into that. And this paper has been published just today. This is the first MRI viral protein, that is nucleocapsid protein, that has been subjected to crystallographic studies, and its structure has been deduced, and it is published in Journal of virology, and it's out just today. So good to see a lot of progress with respect to the molecular biology and the structure of the MRA proteins. And this is fig mosaic virus. The one on the left top corner is the healthy fig leaf, whereas these three are affected by fig mosaic virus. And the virus affected fruits, the fig fruits, they look something like this. And this is quite damaging in the Middle East as well as in the United States and many other countries where fig is cultivated on large scale. And in California, the fig is cultivated earlier, although it is center of origin is in the Middle East, but it is being cultivated on large scale since 1900s, early 1900s. You can see this is the picture from 1900, and this is the latest picture. And these are all severely affected by fig mosaic virus. So it's a very economically important viral disease of fig. And in addition to this, there are certain minor viral diseases in fig also. So when you subject all the fig mosaic virus sequences by phylogenetic analysis, the clustering pattern is region specific. Here you can see all the Israel isolates, they clustered together, and all isolates from California, they clustered together, and those ones from Serbia, they clustered together, and those from Japan, they form a different cluster, and the ones from Turkey, with an exception of California isolate, probably because of mobility of planting material or whatever, they all clustered together. So this tells how there has been region-specific evolution of these MRA viruses across the globe, although they infect the same host. So this is rose rosette virus, which is quite damaging in North America, wherever rose is cultivated on a commercial scale. And exceptionally, there has been report of this particular virus from eastern part of India since last few years. This is quite severe and economic problem in US affecting the rose plants. And there is a lot of diversity of these rose rosette viruses. Hundreds of virus sequences have been generated, and you can see, like the way I explained for fig mosaic virus, the different clustering pattern, the same appears to hold true for the isolates of rose rosette virus, which affects rose, and they show a lot of recombination among different isolates, and also probably segment reassortment. And this is an MRA virus that affect aspen. These are the aspen trees from Europe, that is Norway, Finland, and Sweden. They are severely affected by this MRA virus. Naturally, in many of the ornamental trees and forest trees, this MRA virus is probably quite common. Only thing is, because of poor diagnostic and characterization technologies, we have not been able to identify these MRA viruses. But with the advancement, probably in decades to come, hundreds of MRA viruses would be reported from the wilder the forest regions affecting the tree plants. So probably, yeah, that's about it. And why my slide is not moving? Oh, okay. And all right, this is the genome organization of this particular MRA virus affecting aspen, the ornamental tree. And this is the viral disease in Camellia japonica, another ornamental tree in Japan. This is quite common ornamental tree in Japan, and that's also severely affected by this particular virus. And there are several different strains and isolates of this virus. And again, for this virus, there are two different species. One is called as Camellia japonica associated MRA virus 1, and the other one as 2. Similar is the situation for pigeon pistolate mosaic virus. I'll get into the details very soon. And this is burr. We have burr being cultivated in some of the northern districts of Karnataka, like Bijapur, burr is quite popular. Good that this is not reported here till now, but in China, these burr plants are severely affected by these Emaravirus species, which have generally 6 segments. And yeah, coming to the major problem that is bothering India, especially South India. This is pigeon pea. It looks lush green, but it is not really healthy pigeon pea. This is sterility affected pigeon pea. It has failed to form flowers, and whenever this virus infects pigeon pea plants, it triggers a lot of vegetative growth, whereas it deprives the plant from reproductive growth and formation of flowers or pods or the seeds subsequently. So this picture is from my hometown, and you can see these plants. You cannot harvest anything, although they look pretty good. And you see here a farmer holding a healthy pigeon pea with a lot of yellow flowers, pigeon pea flowers. On the other side, it's a virus infected, although it looks lush green and healthy, but it is a virus infected, sterility mosaic virus infected pigeon pea plants. This virus causes chlorotic ring spots or mosaic symptoms on the leaves, and the excessive vegetative growth and stunting of the plants. It's also called as green plague because it spreads like plague. Of late we have not seen, but probably our elders and the previous generations, they were like COVID. They were really scared of this plague. Entire villages used to be vacated whenever there was outburst of a plague disease. And this is also called as green plague because it spreads as fast as plague in the field. And these aerophid mites will move from plant to plant or field to field, which probably is facilitated by the winds. So what you see here is in the early stage, the plant, if the virus infects the plants, they don't produce the flowers. But if the virus infects the plants in the later stage, although the flowers and pods are produced, but the quality of seeds is really, really bad. And this virus is transmitted by aerophid mites. And you can see the electronic microscopic pictures of the virus particles and a schematic diagram of virus particle. You can see there are 5 or maybe 6 segments, and the largest one being RNA1, which encodes for the polymerase. And you see here, you can see it here, how nicely it has been depicted. So there are 2 different pigeon pea sterility mosaic viruses. The sequences are so diverse, so they are nomenclatured as pigeon pea sterility mosaic virus 1 and the other one as 2. And both the viruses are predominantly present in southern part of India, and sometimes they are also show mixed infections. And there are certain alternate hosts, mostly the legume plants like beans, which this virus gets into these plants either by rarely by artificial sap transmission and mostly by mites. Although these mites are very host specific, but sometimes through artificial means they can be transmitted from too few of the plants. So this is the milestones of SMD research. Now, the first sterility mosaic disease report was way back in 1931 from Bihar. Nobody understood what this disease is really, if it is because of phytoplasma or insect damage or what kind of virus or a bacterial disease. So it was a long journey for researchers to really identify this viral sequence. And in early 2000s, the researchers speculated that this could be because of a tospovirus, because of similar viral structures like that of tospovirus, tomato spotted wilt virus, for which a lot of research has been done. Whereas, in early 2020s, with the advent of the sequencing, next generation sequencing technologies and all, this virus sequence was completely unraveled, and it was kept in an entirely different genus called as Emaravirus, which is a sole genus in the family Fumoviridae. So now we have realized that there are 2 different viral species affecting Emaraviral species affecting pigeon pea. So this is how the pigeon pea virus genome organization is like. There are 6 segments. I already explained, what these first 3 segments encode for. The 4th segments encode for a movement protein, which helps the virus nucleic acid and the virus particles to move from one cell to the other cell via plasmidesmata. And still, there are no clues as to what the segment 5 or 6 would encode for. So this is a northern hybridization showing different RNA segments. You can see all the 6 segments visible in this particular RNA extracted from virus infected sample. So when I was in Delhi in NRCPB, we collected virus infected samples from across the country, and most of them were from Karnataka and some from eastern part of India. And our studies revealed that there are 2 different viral species which are both predominant in entire country in all the regions, and many times there are mixed infections, and such mixed infections sometimes result in recombination as well as reassortment. So I'll tell you what is reassortment, and what is its significance in terms of virus evolution, whether it's animal virus, plant virus, or viruses infecting human beings. So this is a phylogenetic tree. You can see here both the PPSMV1 and 2 are quite distinct. The PPSMV2 viral species is quite similar to fig mosaic virus. We don't know really how this particular virus got an entry into India or how it really evolved, because there is no fig mosaic virus reported from India until now. Maybe future there could be some reports, but we don't know the evolutionary history of this particular virus. Whereas the PPSMV1 is more similar to rosette virus, and this is how these 2 viral species infecting pigeon pea are quite distinct. This is all the phylogenetic analysis of the RNA sequences that we deduced from different samples from across the country, and you can see different clustering patterns. So the isolates from different regions have evolved differently. One interesting thing is you can see here, we developed several RT-PCR and other techniques for diagnostics of this virus, and we developed a multiplex RT-PCR technique to detect the pigeon pea sterility mosaic virus, as well as to differentiate whether the virus type is pigeon pea PPSMV1 or PPSMV2. So this work has been published, and we showed several recombinations within the virus isolates of the same species or across the different species infecting pigeon pea. And for the first time, we showed the reassortment of RNA segments of Emaravirus, and that particularly happens with the RNA4 segment, which encodes for a movement protein, and probably it has a silencing suppression function. And evolutionarily, this is very important because they are pathogenicity determinants. And with this reassortment, the pathogenicity of the virus, evolves along with different reassortments. You can see how these reassortments have occurred in the field, and this can happen only in the scenarios of mixed infections. So thus such reassortments are not uncommon. They have been reported for several human infecting viruses such as flu viruses. Every year we hear about new flu virus, swine flu or bird flu or another kind of flu, and all these evolve because of reassortment, and the vaccines really don't work against these evolving viruses, especially when there is entire segment reassortment. It is not just mutation or recombination. It is because of the reassortment of entire segment which are swapped across different strains or isolates of these flu viruses. So similar mechanism works for Emaraviruses. And it has a lot of implications on evolution of this virus with respect to the virus fitness, virus pathogenicity, and evolution, establishment, epidemics and endemics, and so many other things. So am I closer to how much time I have, Dr. Sridhar?
Yeah, you can take another 10 minutes.
Okay. Yeah, and we did a lot of small RNA characterization for this virus. This is all important because if you want to come up with a biotechnological intervention wherein you want to target the virus by using RNA interference technology or double-stranded RNA-based technology, it's very important to determine the viral sequence which are efficient in production of small RNAs, which are key molecules for control of this virus. So we did a lot of small RNA profiling for different viral strains of this. And this is a break. I have been in IIHR for almost last 5 years, and since pigeon pea is not a mandate crop, I really could not work, but I got an opportunity to visit UC Davis on a Fulbright fellowship. So I made use of this opportunity to again to do some work which has not been done, which is unfinished with respect to the characterization of the viral genes. So, so what I did is, we designed primers to isolate these viral genes and clone them by using latest advanced cloning technologies, by using different gateway vectors and fused to different reporter genes such as green fluorescent protein or its modified versions, and used a lot of vectors. And by transient agroinfiltration method, we did a lot of microscopy work to find out the localization of these viral proteins. It's really important to understand the localization of these viral proteins because that would give a lot of clues as to know what is the function of each of these proteins, where in the cell they are located, and what kind of function these proteins could be doing, and what kinds of plant host proteins they could be interacting with. So this is the nucleocapsid protein. It was present all across the entire cell. You don't see here in the center because it's vacuole, which pushes off the cytoplasm to the periphery. So you mostly see the viral localization in the nucleus or towards the periphery of the cytoplasm. And this is the movement protein, which is mostly present on the towards the cell wall, because it can it does the movement functions, and it's important that it is here, and it helps in movement of the virus from cell to cell across the plasmidesmata. So yeah, we character, I'll not really get into that. And further, what we did was we looked into some of the viral proteins. They move like this rapidly. You can see here how rapidly they move, and it's all important to determine whether they are associated with the cytoplasmic streaming or associated with vesicles or Golgi complex and all that. I'll not really discuss much on that. And we studied the interaction of these viruses, and these interaction studies, they throw a lot of light as to what the segment reassortment would have, implications it would have on the virus evolution. And we saw the proteins encoded by RNA4, which is the reassorted segment, interacts with the several host proteins, and that the assortment is really functional in nature. It is not just that it's more of a mechanical reassortment, but it has a lot of functional implications. So I'll not really tell much about all those things. So this is just to show, it's not really still well organized. This is summary of the interactions. Not all the viral proteins interacted with each other. It's mostly the viral movement protein interacted with most of the other emaraviral proteins like P3, P5, P6. So it tells that how important that the movement protein is with respect to its interaction with other viral proteins or host proteins, and what meaning it would carry with reference to the pathogenicity of the virus and its evolution. So we looked at we did some silencing suppression studies. The silencing suppression was quite weak for this virus. So, all right, in collaboration with GKVK, we also evaluated a double-stranded RNA technology, wherein you express double-stranded RNA in the lab and isolate it and paste put it on the leaf surface of the pigeon pea, and then you staple an infected leaf which is carrying the areophid mites using leaf stapling technology. And then you can see wherever the double-stranded RNA is present, the plant was healthy, not infected by the virus. Wherever this was not treated with double-stranded, the plants were affected by this particular virus. So this is one biotechnological intervention where, although it's more of in a very small scale experiment, but if double-stranded synthesis can be commercialized by using latest advancement, we can use it probably in the field like pesticide or viricide in the field scenario. So we did see some protection targeting different genes. It's all published. Yeah, that's all about these Emaraviruses, and I thank ICAR, NRCPB, IIHR, and Fulbright Fellowship, UC Davis, my colleagues and lab members for all this work which I have described here. Yeah, thank you for your patience and time. These are some of the pictures of my Fulbright visit. I'll just show them very briefly. An Indian farmer in California growing pistachio almonds and a lot of commercial crops. And this is honeybee being kept here to facilitate pollination in these plants. So I had opportunity to participate in several seminars, give talks here and there, visits universities. So it was really a wonderful visit. This is California citrus orchard. You see hundreds and thousands of acres of citrus plantations in just one particular village. And this is the irrigation system. This is an Indian friend who is settled there in California working on citrus diagnostics. He is supported by Farmers Fund, not by government or whatever, but farmers, they have their own labs for disease control and diagnostics. And this is from UC Riverside. So had opportunity to visit some of the industries like Bayer Crop Science and Syngenta. So this is visit to Fort Valley State University and Missouri Baptist University in St. Louis. So I had worked in this particular institute for almost 5 years, developed virus-resistant transgenic cassava that has been released in Kenya for commercial cultivation, and they are working on for its release in Rwanda in Africa. So this is Stanford University. You can see the beautiful campus, UC Berkeley. And yes, I visited several schools, gave talks on viruses and all to the students. It was quite, remunerative experience and visit with this particular fellowship. Thank you. Over to Dr. Sridhar now.
Yeah. Thank you. Thank you so much for a nice presentation, introduction about Emaraviruses. It is quite natural that though you have not covered this management aspect, all the aspects regarding its detection, importance, and how it is spread, everything is in detail you have cleared. I really hope all the participants who are here now they can speak themselves for at least 5 minutes regarding Emaraviruses and how we can manage. Though we are familiar with pigeon pea sterility mosaic virus and vector, regarding this nomenclature and other parts today, it was very clear, at least for me. Others also, they can share. With this, we profusely thank you for your excellent presentation regarding the importance of Emaraviruses. Now it is over to the audience for any clarifications. You can unshare this one. We'll have common discussion, please.
Okay. Yeah.
Yeah, to begin with, I have a few things here. In this group, there are several subject matter specialists, virologists are there, entomologists are there. If somebody is particularly a carologist, that is where this can be taken as one example, where as a group, how we can come out with management. For example, definitely it will be having host plant resistance. You yourself a plant leader, come biotechnologist, pathologist group is there. from entomology, how best you can manage, or with a related science, acarology, how best you can manage these mites, very minute and which cannot fly. How best you can control them for controlling these particular viruses. With this, just I would like to have, because you have already worked for the management of viruses in cassava, you have come out with some varieties also. What sort of approaches we can world over they are following for management of this group of viruses? It can be a common one. Can you share some of the points related to management of this group of Emara viruses?
Yeah, with reference to the Indian scenario, the sterility mosaic disease of pigeon pea, these areophid mites, I mean, you are entomologists, many of you are here are specialists in that area. The acaricide application can help in control of the areophid mites, but my knowledge is from the literature, the publications. So what I understand is these acaricides are not very effective, and as well as probably they have they're highly toxic, or they are not well used by the farmers, unlike other pesticides which target the pod borers and other major pests. And farmers really don't know, they are not aware of these acaricides. So they just go on spraying mostly pesticides like, as per my old knowledge, it's monocrotophos, and I don't know the latest pesticides.
Okay.
So
Yes, because there are several, at least a dozen molecules which are effective for mites.
So that's one. And the late cropping, late planting of pigeon pea, I think that results in a lot of the sterility mosaic disease incidents, because that's the time whenever the planting is delayed, like now the monsoon is late, and because of late planting, the mite population really increases like quite significantly, and that translates into higher mosaic incidence in the pigeon pea farms. The farmers are not really aware, and generally for viral diseases, the major practice is roguing. You pull out the plant if there are a few of them, and you burn them, or the sanitary practices. But probably during that process, since these mites are associated with the plant, you may further facilitate dissemination of these mites to the adjacent plants, and it's quite difficult scenario. So yeah, one thing what I can say is timely planting is quite important to prevent this particular disease. And I have seen some phytoplasma incidents in pigeon pea plants also. Many times this is confused with sterility mosaic disease incidents. So there are some sporadic incidents of this phytoplasma infection also I have seen in my own farm back in Gulbarga. So so I mean, maybe it may take decades or what to commercialize some technique like double-stranded RNA production, and you develop that into a pesticide-like formulation. Because currently the cost, it's not really cost effective. You have to come up with the bacterial strains and fermentation technologies, cultures, and all that, wherein you can produce a lot of such double-stranded RNA extract, and then develop it as a spray, foliar spray formulation. So that's all there. Transgenic, the situation with transgenic, it's very difficult, to get transgenic cotton commercialized in India. It took decades, and now although more than 90% farmers, they grow BT cotton, but it would be difficult to expect such scenario with respect to pigeon pea, because pigeon pea is mostly cultivated by marginal and small scale farmers, not by commercial farmers. So a transgenic approach would not be really feasible. So yeah, but in the West, in Europe and all, the only solution is to use acaricide to get rid of these mites. So that would be the only approach. In wheat in US, this particular Emaravirus problem is quite severe, you know, much more severe than the sterility mosaic disease scenario here in India, resulting into loss of billions of US dollars. Otherwise, this is mostly reported from ornamental plants and all those. It has not taken up an epidemic scale. So before such a scenario comes, we should be prepared with different tools and techniques to control this disease.
Okay, thank you. Thank you so much for giving a brief idea about what are the world-level management techniques being followed to manage this. And now I request others, Dr. Varghese, sir.
Okay, thank you, Dr. Sridhar, for giving me the opportunity. Nice remarks you made and nice presentation. Dr. Basavaprabhu, I hope I can call you Basavaprabhu.
Yes, sir.
And for the, I think in a long time now, I've heard a talk in virology so succinctly and so clearly put. I'm quite happy about that and congratulate you. I would personally like to congratulate you for that contribution of cassava in Africa, and I think that is the kind of approach, an encore of that should come from your contribution in India also, because for the simple reason, from my BSc Agri days, which has been many decades ago, the control for virus has always been roguing, and it continues even today. And of course, vector management. Even in ICAR-IIHR, papaya ring spot virus, you say spray imidacloprid. That's what is told to the farmers. And but that may not be the kind of thing, what we have seen is sequencing these things right from the seedling stage. It's important. For example, the bunchy top of banana, by and large, we have eliminated because we've been able to give healthy suckers and rhizomes. That's one way of doing it. But the other would certainly be getting into a conventional or a non-conventional breeding when we can get some relatively resistant varieties. For example, tospovirus. Tospovirus, we had in 1989 a one-day session in IIHR, in detailed. But even today, they say control trips. That's how after 3 decades, it should come out of that kind of box. So that is one thing I should tell you. RNAi has been in the talk, even in IIHR for the last 2 decades. Nothing seemed to have come and may not come also. But seeing you there, it gives me hope. So I want you to look into that. And another thing is idiophyid mites. Now, idiophyid mites do occur on coconut, for example. See, coconut, we have phytoplasma-like diseases and all that. They say it's root wilt, and you may be familiar. But can it be? It's a loud thinking. Can it be due to the idiophyid mites on coconut? No, because they have not been able to come out with a reason so far. But probably, I don't think, I'm not sure whether they have explored. I was last 3 years a CPCRI RAC member. This issue of idiophyid mite as a vector on anything, causing anything in the palms has not surfaced. I mean, it's a high thinking. There's idiophyid mites on lychee, idiophyid mites on, say, the jasmines. Whether any such things are there or not, we don't know. Or it may evolve over a period of time, some suggestions. Then one more suggestion, what Sridhar has said, I think, I don't know why you are bracketed under social science. Maybe you're a very social being. But
basic sciences.
Oh, basic science, okay. Anyway, okay, basic science, come into the fundamental science. Now, what I would like to tell you, Dr. Basavaprabhu, is, as Sridhar suggested, kindly see if you can encourage the acarologists in the group. I'm so happy you mentioned about Channabaswana. He's my teacher, So, we have in NBR excellent acarology lab collections, and Dr. Sriram is there. And Dr. Bhagyashree, who has joined recently in IIHR, is also having a good background in hackерology. So if you can also integrate with them and have a scheme related. And finally, what I would ask you after, say, 5 years is, what should I tell the farmer? And if you say all this transcriptase and all that doesn't help me, it helps you only. But I need to know the management. I need to tell the farmer what to do. Or should I continue with roguing only? So I know with a person with your such high background, you can come out with something like RNA, which is an exogenous kind of an application. That will help. So these are not really questions; these are comments and remarks. You know, I kind of invite your comments too. Thank you, and congratulations.
Thank you, sir. Thank you. Can I speak for
a
minute?
Please.
Yeah, sure, please.
Yeah, a lot of points, a lot of inputs for future research and all. Yeah, the reason why we were successful in developing transgenic cassava, virus-resistant cassava in the US was, that did not happen overnight. My boss there, Dr. Claude Fouquet, he has been working on that. He spent 13 years in Ivory Coast in Africa to understand this virus disease, and then moved to US, initially in San Diego Scripps, and then to St. Louis Danforth Center, along with Roger Beachy, a well-known virologist who developed first virus-resistant transgenic solution. It's an output of 20 to 30 years of research efforts. And the funding was really consistent. It was not just show of 1 or 2 lab members. There were dozens of technicians who were doing working on an industrial scale, doing a lot of tissue culture, isolating several events, characterizing hundreds of events. So from them, we got 2 or 3 which were really the best, and a lot of collaboration with African countries, CG institutes. So all this really helped. So the bigger question is, if we have to deliver such kind of, replicate such kind of efforts here in India, are we going to have consistent funding? Are we going to have similar kind of networking? Are we going to have similar kind of teamwork, dedication, consistent dedication efforts? Are the people going to work for several years to come out with a product? Everybody has their life planned, and
agenda.
Yeah, it would be really hard to take such risk looking for the future, which we don't know what's going to come out finally. So these are the things. So I have handled several DBT projects. We got first year's money, and later on did not hear for the second years, third years.
Yeah.
I mean, there's so many
issues.
Yeah, it's not easy. If the similar system works in US, probably they would also not be really successful in delivering such products. So, and that has not happened overnight. It's a contribution of a scientist and his group in his entire scientific life career. So that's one thing. And about coconut mites association, what I know is the major virus that is present in coconut is because of a viroid, and the problem, whatever has been reported from southern India, mostly Kerala and all, this is because of viroid, not because of Emaravirus. So till date, there are no Emaraviruses reported to be associated with any of the palms. So regarding chrysanthemum, you said referred to jasmine, mites are associated with the jasmine. Recently I have come across on a research publication on Emaravirus associated with chrysanthemum. So probably in jasmine also similar situation, because most of the researchers, they work on the crops which are important from the point of view of food security than these ornamentals. So probably similar research efforts should help in finding out if there is similar kind of viruses associated with jasmine or other crops. Yeah, and thank you for all your encouraging words, sir. We'll do our best, but we have to see how we are going to perform. I really let us be optimistic and keep working. That's all I can say.
Thank you, Dr. Varghese, for giving points, and Very nice means reply from Dr. Patil. Also, it should be a group activity type. We have our own limitations. We have to make our own pathways to come out. We cannot go on blaming something else. There should be a way to make our own path and go ahead. That way we'll go in future. Dr. PVR?
Yeah, I only want to know, is there any difference in the rate of spread of the vectors being, viruses being vectored by some winged insects like aphids and thrips or whiteflies, and the mites? Mites being relatively poor flyers and they don't, mobility is restricted. So are they less, slower in spreading compared to other viruses?
Let us, there are so many different kinds of mites. Here specifically, I would refer to eriophid mites, which are involved in transmission of these Emaraviruses. Yeah, as you said, sir, these are poor flyers. They are mostly move from plant to plant with the help of winds or breeze or whatever, or movement of people in the field. We spill over these mites on different plants. So, so these eriophid mites are highly host specific, and that is what makes these Emaraviruses, it restricts these Emaraviruses only to those particular hosts. But as you see, cucumber mosaic virus or other aphid transmitted viruses, aphids are not really that host specific. Aphids probably have a lot of broad host range, and that that makes the aphid transmitted viruses to have larger number of hosts when compared to these Emaraviruses, which are transmitted by host specific mites. So that is why the epidemics of aphid transmitted viruses are more severe in scale compared to the mite transmitted viral diseases. However, probably we have better pesticides to control aphids, but not for the mites because of not much awareness about these acaricides among the farmers. So that's, yeah, these are, so aphids or whitefly transmitted viral diseases are because whiteflies, they move from plant to plant. And of late, we are realizing that earlier we did not know that Gemini viruses were seed transmitted. We were of the understanding that Gemini viruses are not seed transmitted, but now we are like tomato leaf curl New Delhi virus. We have shown that it is seed transmitted, and that could be true with several others. Gemini viruses may vary from host to host, or yeah. There is a lot to be done on that. So, yeah, I hope I answered your question, sir, that what makes aphid transmitted viruses spread much faster than compared to the mite.
Yeah, I think still it needs to be, the transmission pattern is different among the mites and our hemipterans. They need to be studied.
Yeah,
that can be. Yeah, yeah. That's Sriram. Oh, thank you, Dr. Patil. Then that's Sriram.
Dr. Patil, thank you for the nice presentation. So for the benefit of the house, most of us we are knowing that Government of India is planning for the Clean Plant program, and in that Government of India is going to invest 1,800 crores. And most of the crop, that is first in the initial phase, they are going to invest only on the crops like pistachio, what he mentioned, or the kiwi fruit and apple, apricot. And I see most of these hosts, they have this Emaravirus problem in the elsewhere. So it is a good message from him that we can communicate to the authorities that while importing from different countries, these quarantine measures, they have to specifically look for these viruses. This is because our purpose is that we have to get the healthy plant material, but those viruses are not in India, especially for these horticultural crops. When we spend 1,800 crores, we have to be very careful. And I have two very basic small questions, Dr. Patil. Other than mite, is there any other way of transmission?
No, sir, not so far. No other insect vectors.
Because for the thrips, at least some transmission, sap transmission, they have succeeded. for the other experimental purposes. Is there any other indicator host for this to maintain the culture, pure culture, other than the perennial crop? Because for the maintenance of the virus in the lab, any other host is there, weed host or the indicator plants?
The major model host that is used for virological study is Nicotiana benthamiana. So far, researchers have not been successful to transmit this virus to benthamiana. One is mites don't affect this Nicotiana benthamiana or the relative of tobacco.
Cuscuta can be used?
No, these viruses are sparingly transmitted to these indicator host plants. But in beans, they have been able to transmit to some extent. Yeah, I really don't know if we can maintain these, use these plants to maintain these viruses as pure cultures.
For the perennial crops, the maintenance of the virus culture is very important.
It's a difficult, it's a challenge. Yeah, so I don't know of any host range studies that are done for perennial crops, but at least for pigeon pea sterility mosaic virus, lot of host range studies have been done and it's published. Data is available. Yeah, it's the transmission is very poor.
Okay.
Yes, sir. Thank you.
Okay, thank you. Thank you, Dr. Sriram and Dr. Patil. There is one online chat question praising this is wonderful presentation, and query is any progress of just let me come back.
Girish is waiting, sir.
Is there
Dr.
any
Girish is
progress of work on virus detection in mite? Somebody from TNAU, Dr. Renuka from TNAU asked. Dr. Renuka from TNAU asks, is there any progress of work on virus detection in mite? Any binding factor identified?
Not much molecular studies in case of mites because these are highly microscopic in nature, difficult to maintain them. These are highly host specific, and the host these mites infest are difficult to grow in greenhouse conditions. So these are all the challenges. So there is not much virus vector studies, interaction studies done with respect to this family of viruses.
Okay, thank you. Dr. Girish?
Girish, thank you, sir.
Yeah, please go ahead.
Yes. Patil, sir, you are telling that that Emaravirus has been reported from chrysanthemum. So is it from which region? Can you tell us?
I'll have to look into the publications. If you probably Google out chrysanthemum Emaravirus, you'll be able to. It should be probably from Europe, I guess.
Yeah, okay, no issue, sir. Now, my next question is, in India, currently, any lab is working on this Emaravirus detection and diagnosis?
ICAR-IIHR is working. ICAR-IIHR is working on. They have been working historically, and even now they continue to work.
Because they are working on pigeon pea, but I am working in the Directorate of Floriculture Research, which is majorly our mandate crops are flowering crops and also ornamental plants. Which you tell as a major source for the Emaraviruses. So any other ICR institutes which are working so that we can have a collaboration in detection and for these viruses?
I think IIPR, Indian Institute of Pulse Research, Dr. Akram did some amount of work.
Okay.
And but otherwise not really much work. TNAU has been working on this sterility mosaic. Some people in Andhra Pradesh, Telangana, Tandur, some regional stations in Tandur and all, they have been doing field-based studies with respect to this disease. But other than sequencing virus isolates and all, I have not seen any major work being done.
Oh, thank you. Thank you for your replies.
Thank you. Anybody else? Last and final call. Anybody else would like to share your experiences or any queries? With this, we are coming to the end of this 154th track. From this association for advancement of pest management in horticultural ecosystem side. I profusely thank Dr. B L Patil for giving a very, very informative talk on Emaraviruses. And it is means equally it is good that he covered all the studies what he did, and it is good to note that because of participation of others like Dr. Varghese and others, now it has become a complete picture. But even Dr. Girish might be having one or two collaborators for his to be initiated work in ornamentals and Emaraviruses. That way, we hope it is an informative talk. With this, once again, I thank Dr. B L Patil to agreeing in a short notice to give this talk and giving a wonderful thing regarding this group of viruses. With this, we'll come to the conclusion, and I thank Dr. Varghese, Dr. TVK Singh, Dr. Patil, Dr. Sriram, Dr. PVR, Dr. Gundappa, and Dr. Shashank, who joined from IRA, from TNAU, from different institutes they joined, and this is what is our expectation. It is next month we'll be reaching a decade of this monthly talk. Next month that also coincides with annual talk of our APME. Last year we have started. First talk was given by Dr. Varghese. Next this month in the July month, that will be the 120th month talk, as well as the second talk, annual talk under AAPMHE. With that, we will meet in the last week, probably in the last week of July. We'll circulate in advance. And another request is earlier there was a proposal. We have covered 154, and next month it will be 155. We have complete list of talks and speakers and emails. I will be sending on a list to all the speakers with a request to make 1 or 2 pages about their gist of their talk, so that we will publish it during the 120th month talk or a decade of monthly talk. With that request, I thank each one of you for joining in this talk and getting benefited. That is the very purpose of making this monthly talks. With this, I thank each one of you, and we'll be with the note that we'll be joining next month. I thank you, each one of you. I'll be ending this talk. Thank you, sir. Dr. Varghese, sir.
Thank you, Dr. Sridhar. Keep going.
Yeah.
Good. Reddy and all. Team in IIHR. Thank you.
Thank you, sir. I'm ending the link.